The overall goal of this technique is to fabricate an array of protein nanoclusters in a supported lipid bilayer, which is compatible with sensitive surface microscopy techniques for cell biology applications.
This method can help answer key questions in the field of cell biophysics, in particular how the nanoclustering of ligands influence T-cell activation and adhesion. The main advantage of this technique is that it is easily reproducible in standard biophysical lab, and it is compatible with advanced surface-sensitive microscopy techniques such as TIRF and RCM.
Though this technique is designed to provide insight into immunology, it can be applied to other cell types with potential applications in cell biology, oncology, and tissue engineering.
To begin this procedure, clean the glass cover slides and observation chambers as outlined in the text protocol. Then, deposit 70 microliters of 2% silica bead suspension drop by drop onto a cover slide held at a 15-degree incline. Flip the glass 90 degrees every 15 seconds for 1 minute while the suspension dries.
It is critical to create a closely packed monolayer of beads and to avoid the formation of multilayers and/or clusters. That's why the concentration and the volume of the bead solution as well as the hydrophilicity of the slide need to be optimized.
After the liquid has evaporated, place the slide inside a radiofrequency magnetron sputtering device onto a rotating table situated 105 millimeters away from an aluminum silicon target. After this, use a turbomolecular